so this will build on some of those things that Rick has talked about, hopefully. And actually, since I am a physician, a clinician, and primarily a biologist, we can see maybe how we have some convergence in what we would actually like, coming from very different directions. Some of you have probably seen this one. So, as a clinician, my primary concern is taking care of patients. And I got interested in doing research because I got frustrated because tools that were becoming available for me to take care of my patients weren't coming online. And this is the US FDA critical path document. It's a decade old now, but it's not changed anyway in terms of its trajectories. And on the right, you can see expenditures on biomedical research, and on left, you can see the success in getting drugs to market. And I'm not the math guy, but those two graphs are not going in the appropriate directions. Right? They should be going together. So this is a gross picture, I'm just going to warn you. This is my manifestation of the clinical problem. Right? So I was a trauma surgeon in my prior life. This is a guy who got stabbed in the neck. He bled out from an injury to a subclavian artery. He had 70 units of blood required in that first 24-hour period. His head is over here, his feet are over here. This is his chest that's wide open. That's his lung. We had to open up his abdomen, so a plastic bag on it so that things would swell up and compress everything. And this is a technical exercise. At one scale, this is trivial, right? Stop the bleeding, fix the hole, give him blood, right? That's not complicated. Now, it may not be easy, right? But it's not complicated. But when I'm in the operating room at 2 o'clock in the morning doing this case, what I'm actually worried about, because that is an autopilot technical exercise, is I realize that it doesn't matter how well I do my job at that point in time, if it works, that he is going to face an extended ICU stay that's going to linger into months, involve multiple systems that are going to go down, subsequent infections, all sorts of bad things that are going to happen that I can't do anything about. Right? This particular trauma to this particular patient has initiated a set of cellular molecular events that, even though I may have some idea what's going on, I have no control over whatsoever. Right? And that is a huge gap in terms of our ability to actually intervene on the systems at the level that we have the knowledge of those systems, or we think we have the knowledge of those systems. And this is one way to visualize this. I deal with organ function. That's a manifestation of disease, primarily disordered organ function. And we know that organs are generated by interactions between multiple cells, and that those cells themselves are the result of multiple interactions of individual molecules. And what we're trying to do when we engineer a molecular-based therapy is saying, I'm going to intervene on this guy, and I'm going to tell you what's going to happen. Right? And I'm going to be able to tell you how that's going to happen across a range of individuals that may have different configurations of those particular molecules. How can I do that? So part of the problem has to do with the structure of biology. Right? It is a multi-scale hierarchical system in which you can't necessarily infer what the behavior at the higher level is going to be by knowing what the mechanisms are at the lower side. Some people talk about complexity, some people talk about emergence. I would consider these to be organizational problems associated with potential nonlinear interactions at a lower level that you just can't necessarily predict what's going to happen at the higher level. So unfortunately, this is our current operational strategy to dealing with that. Right? We do a bunch of research over here. Right? We make a drug, and we hope it works. And I would posit that this is not a robust strategy. Right? This does not really scale very well. So the question is, how can we show our work? How can we decompose this particular process to allow us to do this in a more rational way? Right? So to a certain degree, we've already heard about how you can take big data approaches to the front end of this. Right? Big data is really, really powerful. It can give us patterns, it can give us information about how things fit together, etc. Okay? But it's only part of the answer. And the danger is, it's a trap. Okay? Because if you only think that the data is going to give you the answer, then you're missing half of the scientific process, right? Because the only thing that the data can show you is what might be true. So if we just go back to basics, we have the traditional scientific cycle. This is Bacon, this is Newton, way back in the 16th, 17th centuries, right? Where you take observations, you identify patterns that data, you construct hypotheses, you design experiments that test those hypotheses, you discard the ones that don't work, and you refine your hypotheses, and it cycles forward. And this works, it has worked extraordinarily well for half a million, 500 years or so. But we're having a certain part of a breakdown in this particular process. All right? One is the increased complexity of the diseases we're dealing with, because to a certain degree, we've captured our low-hanging fruit. Right? Even though infections are very complicated, etc., if you kill the bug, the infection goes away. If you have appendicitis, take the appendix out, it goes away. Right? So those disease processes that are linearly decomposable, we can fix those. Right? But the things that we deal with now, cancer, more complicated host pathogen interactions, immunologic difficulties, etc., those are abnormalities intrinsic to those systems somehow. Right? And that's much more complicated. The other problem we have to deal with is this increased dimensionality of data, which on its good side allows us to gain much more insight into the systems we're looking at. The problem is it's led to an imbalance in the number of hypotheses that are possibly generated. So we would propose that the problem that we have in terms of engineering therapeutics is this step right here, right? Because you have to have some inference of mechanistic causality in order to be able to engineer an intervention for that thing. Okay? And no matter how much you process that data, what you're left with at the end of that is a hypothesis, right? And as Rick would probably, substantiate, the more data you have, the greater there's an exponential increase in the possible list of descriptions for that data. What algorithm you choose, how you decide to cluster, blah, blah, blah. Those are all choices that are made. And it does not result in an unambiguous answer, right? You must have some process to go beyond what comes out of that analysis in order to evaluate causality. So the problem we have with the pipeline right now is that while we have mustered computational advances and resources for this process, right, and you can even automate inference as we've seen, the problem is that the testing of that you have to do is still serial. It requires grad students and postdocs growing cells, killing rats, and doing all this kind of stuff, right? And it just doesn't scale. You cannot possibly keep up. Even if you come up with a list of 50 possible things, right? Some guy in the lab has got to decide, well, which one of these am I going to do for the next 3 years? Right? It just doesn't scale. So this is the loop that we've talked about before. So what I'm going to focus on, as opposed to data, is knowledge. Right? I'm going to say all that stuff that Rick talked about is incredibly important and can increase our reservoir of knowledge. But now, after you've got this knowledge, what do you do with it? How do you sort it out? How do you pick? What am I going to believe? What am I not going to believe? How am I going to evaluate this? Right? And the trick here is that every hypothesis you have is going to be incomplete. It's not going to be true. Right? So to a certain extent, you have to discard concepts of ontological truth when you're dealing with biomedical research, because you actually want something that's actually applied. Right? So the question is no longer what is true, capital T true. What is true enough for me to be able to engineer a solution that I can fix somebody with this? And therefore we've shifted the boundary of what our evaluation level should be, and therefore changed this now, the framework in which we're actually looking for. So this is one of my favorite quotes from Borges. You can read this. Right? This is from his short story of the empire of expert cartographers. Right? So what this tells you is that if you create a one-to-one mapping of the thing that you're trying to study, you might as well have the thing in the first place. It doesn't really give you that much information. Here's another example of this. This is from a couple years ago. So Marcus Covert out there at Stanford, he did this computer simulation of the simplest bacteria I believe there actually is. Took 128 computers 10 hours of runtime for one cell division, which I think is longer than it actually takes to divide, and generates 0.5 gigabytes a day. A very admirable technological feat. But me as a biologist, I'm like, what? What am I going to do with that? I can't do anything with that. Right? I don't know what to do with that particular thing. Okay? So the key here is that science is selective abstraction. Right? It is how can we identify some lesser mean of description that doesn't require me to reproduce every single fact associated with that particular object. Right? So this idea of trying to construct basic rules for biology that we're going to talk about, this is based on this. Right? This is the science, this is the physics, chemistry paradigm. What are the underlying laws or the underlying rules? Because the key here is we want to get a pathway to theory. And the key thing to remember about a theory is it's not a possible explanation for how things are. It's the explanation for how things are. So theories actually constrain the world of possibilities. What we want to do is cast away implausible dynamics, implausible mechanisms, implausible configurations. The theory does is it constrains that. Right, so the problem with biology is that we have this empirical history of studying things in incredible detail. Right, so my system is different. You hear this at every biology conference. Oh, my cells are different. My lab is different. My disease is different. Right, my organism is different. So you run into this problem of infinite regress, where you just give me more data. Just give me more data, and then I'll have the answer. Just give me another experiment, a deeper characterization, more genomics, more proteomics, whatever. Okay. So, but the questions of science are what, how, and why. So if we break these down into individual components, we can see what fits in one particular section. Right? So the output of the scientific inquiry is knowledge. How do we get to that particular point? So the what of biology you've already kind of talked about, right? It is the data, it's the description. What am I dealing with? What is it made up of? What is the description at that particular level? All data goes to defining a phenotype, whether it's a gene expression, it's a list of molecular profiles, etc. That is a characterization of that particular organism at that particular time. Now it's generated by the interaction between its base genome and the environment, but whatever data you collect represents a phenotype. And this is agnostic to scale. So I can look at a patient and say, all right, he looks bad, these are his vital signs, this is his blood gas, this is his gene expression profile. All those are phenotypic descriptions, and you can just throw a big statistical correlation thing on it and come up with an answer, a prediction, which is statistically valid. Unfortunately, it's not valid, one, to engineer anything about it, or two, for individual patients. So what we actually want is to get to the how of the biology. The how is the mechanism. How does one phenotype lead to another phenotype? So this requires intrinsically a dynamic interpretation of the data. So now you're not talking about snapshots, but you're talking about movies. How do I go from one particular picture to the next particular picture? And this is important because mechanisms don't necessarily commute. Right? The sequence of events matters. Right? So now it's no longer scale agnostic. Now you're stuck in that egg diagram that I talked about, and we're able to generate the higher level phenomenon. You've got to figure out mechanisms. Right, so now we get into the issue that's a little bit dicey, is why. Right, so this is previously taboo because it's got this vitalism, teleological thing in the back cast, right? But why is also the pathway to theory because it suggests a meta process. So I would argue that the final cause of biology, the thing that binds all biological systems together, is evolution. That's the one thing that we can say with absolute certainty in our observed world. This every biological object we see is the byproduct of evolution. And therefore this is biology's theory. Right? So if you're going to bind things together, you need to take into account evolutionary principles, and those are diversity, fitness, selection, and persistence. Right? So when we think about biological systems, we have to come back to these particular concepts. Now, how do you execute that? That's the challenge. Right? So we propose that modeling simulation in bioscience is a pathway to theory. Right? And instead of just trying to build the perfect thing at the beginning, we understand it's an iterative process, and when we use the successive tiers of validation that the modeling simulation community uses in order to progress this and forward. So face validity must come first. It must behave in a plausible fashion, and this shifts what we would expect out of our simulations. So the metric is no longer, can I predict this with X amount of accuracy? The question is, does it behave in a plausible fashion? And the reason is that biology is a discovery process, not an engineering process, because we don't know what the system works like, right? So we can't establish the constraints, and we can't operate within a constraint boundary, right? So there's very, very many similarities between what we're talking about between engineering and biomedical science, right? But the main difference is that the ultimate target of this, at least at the outset, is a discovery process. We don't know. So we talked about the importance of dynamics a little bit, right? This is the key that the system evolves over time, that applies mechanisms, etc., right? And what we need to be able to do is to capture that movement of health and disease as a change in dynamic state. So it's not something different, it's a different state of the same thing, right? And that's different because that implies that there's an underlying structure and process that is somehow being modified to generate those particular phenotypes, right? This is a basis of similarity between how you get the particular phenotypes. So the basic insights you get are that the healthy state is a dynamic steady state, and it's actually very, very hard to get right. So you see a lot of disease models that are models of disease, but you can't actually get back to the healthy state from those disease models because they don't model health. Right? So if by visualizing disease as a perturbation of a healthy system, it gives us a reference point to which we can transfer back to. Right, so you guys know about these different kinds of computational modeling. I don't do math, so I do agent-based modeling, which most of you have actually heard about, which is an object-oriented, discrete event, spatially explicit, rule-oriented modeling method that takes a system, breaks it down into components, and has them interact with each other. The key here is that I use it for dynamic knowledge representation. Right? In other words, I have a bit of knowledge. I'm not going to ask you why you believe that. I'm just saying this is what you believe. And you are going to see how that knowledge behaves if you make it go. Right, so most people have seen something along this line where this issue of population dynamics influencing how the behavior of an aggregate system actually works. When they first made the Lord of the Rings CGIs, they couldn't get the armies to fight, because they programmed what they thought were very reasonable stand-and-fight rules, but every time the armies came together, they turned around and ran away. Right? Because they could not have predicted, based on the individual mechanisms, how that aggregate population was going to work. Right? So you see very many mirrors to biological systems in that fashion. So the hope is that you can use agent-based modeling or some similar type of method to essentially reconstruct everything you take a part of with reductionist biomedical science. Because you have to do the reductionist stuff. If you don't do reductionist stuff, you don't know what things do. And if you don't have those mechanisms, you have nothing to build upon. Right? So this is a way that allows you to reconstruct in some way what you think is actually taken apart. So I'm going to give you two quick examples. Right? One has to do with a disease process called necrotizing enterocolitis, which is a disease that affects neonates, premature babies, when you feed them. It involves prematurity, it involves intestinal feeding, and it involves some bacterial components, but not a particular specific bacteria. Right? So the problem is when they study this in the lab, they do sorts of things that are not real in the clinical setting. So they throw little baby mice into refrigerators and hypoxic chambers and all this kind of stuff in order to induce the phenotype. And the fact is that that pathway is not the same as you would see clinically. Right? So the question is, taking what we know has to be present, the necessary components of necrotizing enterocolitis. How can we come up with a minimally sufficient hypothesis to describe this? So what we had to do then is tie feeding somehow to inflammation. So we came up with the idea that immature lining of the gut was particularly sensitive to oxidative stress damage from feeding. So normally when you feed us, right, we undergo oxidative respiration, we generate our ATP, but we also produce some reactive oxygen species as a result of that. Under normal circumstances, our ability to manage that is perfectly capable and never becomes an issue. But what if that was immature, if you had a premature baby? Right? What happens if that is enough to make your cells fragile so that they become easily damaged and can set into a cascade these events that would eventually lead to necrotizing enterocolitis? So this is the standard diagram, right? That's slide 3 in most talks, which shows you all the things that are connected to each other. Right? So we have goblet cells, and enterocytes, we have abstracted blood vessels, etc. We have abstracted bacteria on the top, and all these sorts of pathways and stuff that happen inside. This is done in NetLogo, which is like a child's toy, but really, really powerful in terms of doing agent-based modeling. And the idea is to try and characterize what these host-pathogen interactions are, right? Not to give you a specific gene or a specific protein profile, etc., but essentially to establish what the behavior space might look like of the possible functional characteristics that the components actually have to have. So what we did was we simulated this, the existence of bacteria with different virulence characteristics. We're being agnostic with respect to what those bacteria actually are, but we know that some bacteria produce these types of things. Right? They produce something that breaks down mucinase, which increases encounters with the epithelial surface. Some of them make cytotoxins, some of them have both. Right? And the question is, what does the disease space look like? Because if you can characterize the host side in terms of genetic predisposition based on gene expression levels of their reactive oxygen species managing systems, and you could characterize the microbial community in terms of those things that have the potential to produce these virulence factors, you can identify risk for an individual neonate of developing this particular disease. The other thing that this does is it gives you a finer granularity in the characterization of the disease. So for instance, not every patient we think is going to have NEC has got NEC at the time that we think they're going to have NEC. Right? So there's this transition zone, this NEC scare phenotype. Now we don't know what to do with these. Well, we do. We just stop feeding them and give them antibiotics. Right? But some of them progress and most of them get better, but we can't characterize what the differences are. Right? So this model helps us identify what the components are that may tip you over and render standard therapy ineffective. So I'm going to show you a little model that we've been working on the last couple of years that's a little bit more sophisticated. This is a virtual gut model that looks at the histology, that reproduces the histology of the gut and combines inflammatory modules with morphogenesis modules. So the supposition here is that many intestinal diseases are characterized in terms of histology. Right? And one of the benefits of agent-based modeling is since it's spatially explicit, you can generate spatial pictures. So we know what pathways are involved in tissue patterning. Those are morphogenesis pathways. Well, we also know that inflammation is involved somehow. So the question is, we adopt this modular control structure concept, right, to be able to compose a model of the different components that are there. And the question is, how can we construct a general model that if you perturb in different ways can generate multiple phenotypes? Again, this goes back to the idea of trying to find some underlying commonality upon which you can build. Right, so these are the pathways. Inflammation over here, morphogenesis over here. And in actuality, these two pathways are not connected. So we hypothesized, or we found some evidence, that this gene P10 might be the link between the two. We instantiated that in this particular. Right, so segment maps histology. This is histology of the gut. Right? It takes a topological structure like this, rectangular prisms, and then you unfold the prisms. this model. This is implemented in Repast for the initial implementation. So basically what you see is the crypt layer here, enterocytes migrate from top to the villi, they slough, they fall off, etc. And what we're looking for is changes in inflammatory or mediator milieu that will change the balance between the crypt depth and the villus type, which are histopathological characteristics of certain diseases. We have a high-performance computing implementation of this model. This is a screenshot of that particular model. So we now have a 3D rendering of those rectangles. Here you see the little cells, and it looks very similar to what EM of what the looks like.
Here is it. It's the relative aspect ratio. They're approximate.
Yeah. The idea is to do the rendering so it looks as much like the biology as possible. Biologists are very visual people. You show them a pretty picture, they're like, ooh, that's pretty. All right, so they like that, right? Look very similar to what they're used to seeing. Right, and along those lines, we calibrated against mediator gradients for these morphogens. And we manifested three disease conditions. So local injury in terms of damage to the villus. This is consistent with either a surgical injury, a biopsy, or an anastomosis. We also simulated ischemia reperfusion and validated the hypothesis that enterocyte sloughing in response to ischemia is protective to ongoing tissue damage, and looked at metaplastic change. This is in patients who have Crohn's or have ulcerative colitis who have remedial surgery for that. In their rectal pouch, they develop this alteration in their histology where they get very deep crypts, very shallow villi, and become prone to inflammation. Yeah, so what does all this get you? You do all these models, it looks very cool, it's kind of nice. You can do one particular experiment. But the question is, and this goes back to what Rick was talking about, is that what you want to do is instantiate thought experiments. Because remember, we're dealing with knowledge here, right? And I'm not actually making a claim as to why you believe what you believe. I'm just saying, if you believe this, this is how it behaves, right? So if it looks plausible, then you start trying to break it, because that's how you actually advance knowledge, is by breaking your hypotheses. And if it's already broken, then you go back and reassess your assumptions and see how things are actually working. So just be careful, right? You get in much more trouble by thinking something that's certain than not knowing what's going on. Ignorance is better than being wrong. So what we want to be able to do is to limit the possible interpretations of data. This is the theory stuff. And the fact is that there's no shortcut. Right? You can't circumvent that scientific cycle. If you want to engineer therapies, you're going to have to get down to mechanism. And the question then is, how can you actually scale that cycle? So we have a proposal, right, that we kind of fit in with what Rick was talking about in terms of all the large bioinformatic tools, which is something called the computational modeling assistant. Right? Because the concept here is that if you can't do this plausibility testing in the real world, maybe you can do it in silico and see whether or not, given a particular hypothesis structure, its space of behavior actually fits with what is observable. So the idea here is that you have a biological concept extracted from the data, however you get this, and since we're dealing with biologists, we don't want them to actually write code. We just ask them to express their biological hypotheses in something as close to natural language as possible. And basically, we have just a list of propositional statements. Right? What the CMA does is it says, given this hypothesis structure, what possible forms of computational models can be made from that? First of all, is that hypothesis structure internally logically consistent? Right? That's the first question. And second is, if it is, how can it be mapped to the existing suite of modeling tools that are out there? Right? So what we've done here is done a test case with respect to the virulence activation of a bacteria called Pseudomonas aeruginosa, right, where we kind of know what those quorum-sensing virulence pathways look like. And we had me write down all this stuff, and then the person who developed this, Scott Chrisley, who works with me, put it through this interface into it's an AI that uses MAUD as a logical rewrite language, to see whether or not it can be actually mapped to various types of computational models. So you can do a continuous spatial PDE. It knows it's the PDE because you're talking about diffusion, therefore space. Or you can do a discrete feature net type of model. Right? And it uses internal XML format that's compatible with standard markup languages. So the idea is that what the CMA actually does is it does logical mappings between your biological statements, and then a knowledge base that contains rules for mapping what you need in order to create certain types of mathematical models. So mathematical model specifications. So it will generate, one, a specification of that particular model, and two, it will generate sub-optimized code, right? So you can actually run a simulation experiment. Now, obviously the devil is in the details because those mappings don't exist, and that's one of the things that we're working about. So you can do ODEs and PDEs relatively easily. You can do discrete models relatively easily, logical models, etc. But these agent-based models are much more difficult. So since there is no underlying formalism for an ABM, we're probably going to map to some well-accepted ABM modeling toolkits, etc. And we understand that the resulting code is not going to be optimized, right? But once you've got to that particular point, that's a huge leap. Because what you're doing is you're essentially taking a year and a half's worth of computational biologist time, programming time, and you're condensing it into an automated, semi-automated process. Right, so this is important because actually at the end point, we have to leverage evolution for our scientific ideas. Right, so you're going to have all these correlations, all these data-driven techniques to identify sets of plausible hypotheses, and then you're going to have to run those hypotheses in parallel in some way. So that needs to be instantiated, it needs to be executed, and then distributed across the community. Because, as any of you who have dealt with biology knows, the ambiguity inherent in biological research is huge. Huge, 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 huge. So that was the last slide. Any questions?